It looks like most of the seats are filled. Let's go ahead and get started. I'm sorry about this room and the seats. They're absolutely horrible, but that's part of PSU, unfortunately. And I'm sure there are way better ones elsewhere on campus. But yes, this is virology. Everyone here is comparative Middle Eastern religion? Wrong place? Okay. Good. Yeah, I actually tried to get this moved over to SB1, room 107. Unfortunately, was not able to do that. So, yes, this is virology. Where do you find me? Basically, room 466, SRTC, the building previously known as Science Building 2. Office hours are right after class, and that'll be, come and grab me here. Virology. No. Scared one away already. Cool. So, yeah, right after class, come and grab me. Again, immediately afterwards is probably the best way. I don't know if there's another class in here afterwards. Often there is. How many people have class right after this or have to get down to SRTC? Not SRTC, the CLSB. Too many acronyms, yeah. So for those of you, I'm more than happy to set up other office hours by appointment. As long as I can do that. I'm teaching quite a bit this term. By far and away, those of you who've had my classes before know the easiest way to reach me is by email. I'm kind of email dependent, and I try to respond to it pretty quickly. You can try and reach me via D2L mail. I may or may not notice that the icon is blinking up in the corner. So just send me email to my normal PDX email address. If you want to talk to my voicemail, go ahead and call. But that's about it. A lot of people have asked me about prerequisites for this course, particularly having to do with cell biology. How many people taking cell this term? A whole bunch of you. Okay, so, or haven't taken cell biology, sorry, or are taking it this term. Okay, I know a few of you for that's the case. But how many have not had molecular biology? And I don't know your names, so that's fine. Anybody not had molecular biology? Good. Because if you haven't taken it, this class will be really challenging because this is really a molecular virology class. Very little about pathogenesis, and I'll talk about that before. Why is that? I'm just going to assume that you've had molecular biology and some bits of cell biology. The parts of cell biology we're going to be talking about are cell surface receptors and intracellular compartments. And hopefully most of you have had that in sort of a principles biology class. So that should be pretty straightforward. If you have any questions about this right after class today is always a good time to come and find me and ask about that. As I mentioned before, this is really a molecular virology class for a couple of reasons that I'll get to a little bit later on. And the main classes of viruses, there are really sort of two ways to do a virology course. One is actually kind of the way we're doing it, which is each of the individual viruses and different virus classes. And I find it's easier to sort of compartmentalize that way. Another way to do this course is to start with replications, entry, etc. It's another way to do it. It's in fact the way that this textbook is put together. Really nice textbook. This will be on reserve in the library, by the way, as soon as I'm done here and done chatting with you. But so we'll literally be talking about the individual different classes of viruses and usually just the one or two examples from any of those given classes. So 3 main classes of viruses, the DNA viruses, the RNA viruses, and the retroviruses. And hopefully when we have our guest lecture, he'll try and convince you there's a really cool different one kind of in between that we work on in my lab. Everything from bacteriophage and our friend bacteriophage T4 all the way through to HIV, and then maybe some of the other interesting ones here. A lot about this course, and again, kind of why I hope people have had molecular biology beforehand, is this course is a lot about where everything you learn in molecular biology is not quite right, or at least how viruses do things differently. Although, to be perfectly honest, a lot of what we talked about in molecular biology, and by the way, every time I say that, people make little notes and say, when we talk about this in molecular biology, I think I got to 51 in one class. So it's really about how we know a lot of things about how cells work and how differently things are working in viruses. So again, a lot of the fundamental aspects of cell biology, molecular biology we know from the study of viruses, and then the weird differences. And it's sort of the weird differences the focus of this course. We'll talk a lot about genome replication. How do you make more of the virus itself, particularly its genome? The rest of the virus, which is assembly, putting it together. And also look at gene expression as how you make the eventual proteins which you need to make all the rest of the virus. I'll talk a little bit about pathogenesis. Pathogenesis is probably kind of like me. I have a little bit of a runny nose right now. I also have 8 hours of jet lag since I got back from Europe last night. So I was joking with my 10-year-old daughter as, being infected with the virus is kind of like having jet lag. So general malaise. So we won't really talk much about pathogenesis with a couple of exceptions. Influenza is probably sort of the main one of that. Unfortunately, it's not just all about me chatting about viruses and us discussing them. There are a couple of exams as well. 2 midterms and a final. The midterms Wednesday, 22nd of April, and Wednesday, the 13th of May. There's a non-cumulative final, basically like a third midterm, on Tuesday. It's the regularly scheduled exam time at 8 a.m., not at the normal exam time. All exams will be 50 multiple choice questions with Scantrons. With the blue form, there are lots of examples of old exams on D2L. I've posted pretty much everything on there. You will notice that 30 plus 30 plus 30 equals 90. So what do the other 10% come? These are the clicker questions. I will be doing daily quizzes. I will have them first thing at the beginning of class. If somebody absolutely has to leave early on, please sit on the edge of the room so you can get out easily after that. Any type of clicker works. iClicker 1, 2, 3, etc. Unfortunately, they all have to be iClickers. Anybody not have clickers? You will need to get some. Any questions about it, let me know, and we can deal with that. That being said, you can get out your clickers now, because we'll have one non-graded clicker question. This is more for me than anyone else. It makes no difference whatsoever in your final grade, but it helps me know sort of the distribution of people that we have here, and may actually end up getting us better resources in the biology department if we can figure out and tell people what's going on here. So now we can actually start our clicker questions. Now everyone's got them out.
What is your more than one of the above?
More than one of the above? I would just say pick the one that you most closely identify with. So junior biology major, senior biology major, post-bac, junior, senior, non-biology major, or none of the above. And I always give a minute and 30 seconds for clicker questions. So, and then counts up here. This is, in fact, I'll probably be counting down the next time around because I guess I didn't recheck it for that one. And are you guys interested as well in what the results are? Yeah, well, I'll show you what. Again, I don't know. So far, we have none of the aboves. Does nobody want to admit it? I'm certainly none of the above. Okay, anyone else want to vote? No? None of your clickers here? You don't want to share? Okay, fine. We'll just go ahead and stop. Show results. So most of you are seniors, 67, 14% are juniors, and 19% are post-bacs. So that just gives you a bit of an idea distribution what we have here. Since, of course, I don't know who you are yet because you haven't registered your clickers, so we can need to register them. I'd use the website for clicker registration. Please do it by the midterm, name, clicker number, and your Odin ID. That's the way that D2L keeps track of things, and so I will do my best to upload clicker scores to D2L on a regular basis. Having the Odin ID makes it a lot easier than any other way of looking at it. What kind of grade will you get? Any of you who've had my classes before know that my exams are ridiculous. It's one word that's actually a really nice word that people use. Usually people use not so nice words about them. And because of that, I set the top score to 100%. And then I scale from there. The top scores on my exams are usually in the mid-40s out of 50. And the mean is usually at about 25 or 26. So consider yourselves warned at this point. I've tried writing easier exams. It turns out the median is lower. So I guess that's it. Exams, we have to go through this anyway. They're a closed book, no time limit on taking exams. So if you are going to the testing center just because you need more time, that's not a problem. We can stay here absolutely as long as you need. Then we can go outside. We can go over to my office as long as you need. I've done the calculations, actually just plotted how long people take and how good their exam scores are. It's a flat line. There really is very little difference on average. For you, it may end up working a little bit better. Everyone gets to keep their exams when they leave, which means once the first person has left, they have the exam. And so I can't let anybody start after that. The same thing goes for the testing center. So it has to be at the same time. So if you are planning on taking your test at the testing center, sign up now, particularly for the final, because last term the final was completely booked out all day. So sign up for those immediately. And also what that means, of course, is be here on time on exam days. I'll let you read this. I don't require attendance. You could say, okay, 10% for your clicker scores. But even without clicker scores, if you always come to class, you're generally going to be getting a better score. So if you see here, people who've self-reported, admittedly, always come. There's no guarantee you'll get an A or a B, but most of the people do. You can get an A if you come very rarely, but you're generally going to be a lot better off if you always come to class. And I've also asked pretty much all the people who've gotten high scores in this class in the past, also in my molecular biology course, they say, come to lecture. Come to lecture, come to lecture, come to lecture. And as we'll see a little bit later on, you'll notice I'm mic'd up. I'll be posting all of the lectures, all the audio and video on YouTube. And so checking those out as well, most people seem to think really helps. If for any reason you really, really, really can't be here, there are going to be extreme conditions, and it's an essay exam, and people who've taken them before haven't been really happy with them. Sometimes scantrons end up being misgraded. I give multiple different versions of exams. Sometimes the scantron will be scanned on the wrong version. So let me know, but let me know within a week. And I will be scanning those. So how to do well in this class. You don't absolutely have to have the textbook. I will be putting it on reserve in the library, but I strongly recommend it, and it's actually not ridiculously expensive. This one is about half the price of this one, which is one of the other reasons to switch. Second edition of Fundamentals of Molecular Virology. There are a couple of things in here that I really like. There's chapter overviews, the beginning of each chapter, which are really nice reviews for, say, exams. Just sort of going over the various different aspects of what's here. Also, a glossary of key terms at the back. Two very good things to be looking at in terms of revising what's going on here. There are also really nice references if you want to get into any more detail. I'm usually not going to be getting into the detail that any of those original references are in. But if you're interested, and hopefully you'll be interested, you can go and take a look at some of those. D2L, how many people got the lecture notes for today already? Good. I try and post them by 10 o'clock the night before, assuming that I'm on top of things. Sometimes it ends up being a little bit later. All of the previous year's lecture notes that I'm kind of basing these ones on are there as well. Last year's lecture recordings are all on YouTube. Probably the easiest way to get there is just to subscribe to my YouTube channel. And that way, anytime I post something, it'll be updated there immediately and you'll get an email. Anyone who's had my classes before know that I talk too fast, and the best way to slow me down is to ask questions. And if you have a question, there's probably somebody else in the class who has a similar question, if at all. What's on D2L? Syllabus, all the lecture presentations, old exams. Most of them have keys. Bug me, and I can also post exams that don't have the keys that are already listed on them, so you can take practice exams. I always think practice exams is a very good idea. There are some supplemental materials. If you've looked at the syllabus, you'll notice a couple of places have some extra readings, which I have posted on D2L. Those should all be there. Discussion forum. I try and read this on a regular basis. I set it up sort of with questions and answers. Somebody sends me an email, because again, that's the best way to reach me. I'll post the question and answer. I usually anonymize it on D2L. And so those things are there. So again, because I feel that usually if one person has a question, there are usually multiple people who have similar kinds of questions. Also, people use the discussion forum to set up study groups. I highly recommend that. I know that people have had good luck with that in the past. I won't be reading that section of this D2L. I do occasionally check my mail on D2L, but as I say, you're much better off sending it to my PDX email. That's only one place where there are resources. I mentioned Principles of Virology before. This is the second edition. There's a third edition out as well. It's a first edition. This will be on reserve in the library. This is great for animal viruses, and that's in fact one of the reasons that I decided not to continue to use it. It doesn't really cover any of the vast majority of viruses that are present on our planet. And so that's why I decided not to continue to use this particular textbook. I've been talking to one of the authors, and there may be, in the fourth edition, now some more of the non-animal viruses which are there, but I doubt there's going to be that much of it. So again, this will be on reserve in the library as soon as I get over there after class. Fields Virology is sort of the holy book of virology. It's, I think, now in 6 volumes and covers all viruses in more detail than you ever want to know. Partly the reason there are 6 volumes of this textbook is that things are being published about viruses all the time. Journal Virology, Virology, Science and Nature. You have access to, I know, Journal Virology, Science and Nature. I'm not sure about virology directly. Anytime you're on campus, you have the IP address that's here. You can get access to all of those journals. And some of the stuff I'll be showing in class will be there. The Stedman Virus Library. Anytime you try and go to the library and check out a book on viruses, some other person has probably checked it out, which means it's in my office. So come by and you can check those out on a relatively short-term basis. One of those is this book called Life in Our Phage World, all about the viruses that this book doesn't talk about. Supposed to be a field guide to what has Earth's most diverse inhabitants. I wrote one of the chapters in here. It's really a very fun book to take a look at some point. And of course, office hours after class and whenever you can schedule them. This is a long list of incomplete virology resources which are out there. Virology.ws is a blog by Vincent Racaniello, and those of you who are up front may have noticed that the third author on here is Vincent Racaniello. He likes to call himself the world's virology professor. Small ego, but is really good. I really greatly appreciate the stuff that he does. He's in fact part of the inspiration for me recording my lectures and putting them online, because he does that for all of his lectures as well. And you can connect to all of those lectures there. He's at Cornell. He also has a Coursera course on virology, and some of particularly the first lectures there I think are really good. I didn't get much past that. He also does a podcast with some of his buddies is a little bit long. It usually ends up being about an hour and a half to 2 hours. And I've been on it. I was on the guest at one point, and then have submitted a couple of letters as well to that. Usually really great, and they've done this wonderful section. I think they had about 2 months on Ebola and various different aspects of it. Another couple of interesting places, the ViperDB, which has a bunch of structures. One of the really fascinating things about viruses, and one of the things that makes them really different from all other life forms, as I call them. We can argue about whether viruses are alive later over some alcohol. But the structures are really unique. The virions, the virus particle, has really, really unique structures. And so one of the places to find that is at this ViperDB. Many of you have been subjected to this movie that I'm working on. This is The Edge of Life, and I'll show you the trailer if you haven't seen it before. If you've seen it before, you can turn off for those 4 or 5 minutes, probably be towards the end of the class today. But there's also a website for the movie. It's a Facebook page, just this particularly links to it. I post on there what I think are interesting virology stories, particularly a lot of input now recently on vaccines, the whole measles outbreak, and that kind of thing. So I put that there. Viral Zone has really nice overviews of virus families. This textbook, in the first couple of chapters, is also split up into various different virus families. So it's another way to get information on each of those individual virus families. You do have to be a little careful. I looked at the section on the viruses of Archaea, which is what we work on, and there's some really big mistakes in there. So free is wonderful. And then this book, The Centennial Field Guide to Earth's Most Diverse Inhabitants, came out of a birthday party. So 100 years of bacteriophage, first described in 1915. This link actually takes you to the party that we had in January of this year, where you can also see all the talks, again, all available free online, including one about archaeal viruses that I gave. So full disclosure on all of this. What are some of the cool things, other cool things we'll be talking about in this class, depending on whether we have any graduate students who snuck in at the last minute? Looks as if we didn't. We'll have an extra lecture at the end on bacteriophage T4. I don't know why they didn't have a chapter on bacteriophage T4. I'm guessing the person who was supposed to write it bagged or didn't get back to them on time, because it's such a fascinating virus. Visiting lecturer on the 15th of April, George Kassoon, who's a PhD student in my lab, will talk about some of the research that he's doing on some of these RNA-DNA hybrid viruses. And then on the 17th of April, there will be a lecture on Lambda, but it'll be through a video DVD. And the reason for that is this letter, which I have here, from the University of Jyväskylä, which is in the middle of Finland. I didn't know where that was. Faculty of Mathematics and Science appreciates your promise to act as an opponent at the dissertation of XYZ. Dissertation will take place on the 17th of April at the University of Jyväskylä. So that's where I'll be. I'll be in Finland on the 17th. And to get there, you have to leave like insane amount of time early to actually, I didn't realize quite how far away it was. And this, this is the procedures. The procedures for the dissertation are this long, and I'll just read the first one here. So the doctoral candidate, Kustos, the chairman of the doctoral dissertation, and opponent should all wear similar types of clothes. Men should wear either a tailcoat with a black waistcoat or a dark suit. Women should wear a black long-sleeve dress with a small neckline without a hat. All can also wear the ceremonial gown of the university. The doctoral candidate chooses the style of clothing after talking to the Kustos. The opponent can also wear the ceremonial gown of his/her home university, not finish. The use of ceremonial gowns, etc., etc. In the beginning of the public examination, the doctoral candidate arrives into the hall first, then the Kustos, and then the opponent. The audience remains standing up. During the entry and exit, the custos and the opponent hold their doctor's hats, if they have one, in their left hand so that the liar is facing forward. So that's where I will be on the 17th of April, carrying my hat in my left hand with the liar facing forward. So we will also talk about some of the viruses we work on in my lab. I mentioned before, George is doing his PhD on what we call the RNA-DNA hybrid virus, which is a really fascinating genome we found recently. We'll talk a little bit about SSVs, which is the sulfolobus spindle-shaped viruses. Again, the main work that we're doing in my lab, but also what the recombinant DNA techniques lab is going to be working on as well. Talk a little bit about STIV, which is this virus up here in the corner. I should say this is the virion up here. This is a virus that I discovered in Yellowstone National Park about 15 years ago now. Towards the end of class, and maybe some of these viruses from the Stedman lab, we might do a flipped lecture. Everyone know flipped lectures? You watch the YouTube video beforehand, then you come to class with questions, and we go from there. Interest? Yes, no, good, bad, give it a try. Some of you may have heard about this virus recently. We'll talk more about Ebola when we talk about filoviruses. Some of the craziness which is going on. Is it done? Is Ebola gone? No. Still happening. I was going to give a situation report. Probably one of the best followed viruses that are out there. Any ideas how many more people will die in the US this year from influenza than have died from Ebola? About 20,000 more, maybe 30,000 in an average year. And then we'll talk a little bit about measles and the whole measles epidemic and why vaccines are a really, 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 really good thing. I always like to say with influenza, anyone who's sitting within a meter of you is exposed. And these seats are about almost exactly a meter apart. So I've measured them. Measles, everybody in this room, if one person had it, would be exposed. And probably the whole campus. So that's one thing to do there. So any questions about how the course is going to run at this point? As soon as I'm done with this, I will, again, be posting things online. Was there a hand over here? Questions, comments, worries? Yeah?
How long would you say you follow the book?
Good question. One of the things you'll notice if you look at the syllabus, it has chapters in there. And so I try and follow those. You will also notice if you look at the syllabus that they're not in order. So I do jump around a little bit. These are things that I think fit together better. So for instance, the lecture, this guest lecture on single-stranded DNA viruses and what we've been working on in our lab kind of bumped a little bit. The viruses of Archaea, I moved all the way to the end. But for the most part, it follows the textbook. In terms of the material which will be on the exams, and people always ask me this, I don't go back and look at the textbook. I go and look at my lecture notes. But they're pretty closely based. You'll notice that almost all of the figures that I have in my PowerPoints will be from here with new ones that are added. But pretty much every single one from here will be in my PowerPoint slides. Other questions? Yeah?
What is a doctor's hat, and do you have one?
What is a doctor's hat, and do I have one? This is the, it varies, but usually they're 8-sided and with a little tassel on the end, also known as a tam. But where the liar is, I have no idea.
Isn't that whoever loses the debate?
Yeah, well, something like that. The thing I didn't mention is that this is a public debate up to 3 hours with just me asking questions and the PhD candidate answering them. Yeah? Is that going to be filmed? Not to my knowledge. But say I have the dubious honor, if I'm not sure it's the right term, of being one of the world's experts on viruses of things that grow in very high temperatures and extreme environments. And so that's why I was invited to do this. Do I speak Finnish? No. In fact, somebody up here was asking me a little bit earlier, so I just got my train tickets to get from Helsinki up to Jyväskylä, and they're all in Finnish. And I think there's a reserved seat on there, but I'm not quite sure where it is. And the translation from Google Translate was not very useful. But apparently, I've heard that everyone there speaks English probably better than me. So, any other, I will report back when I get here, and I'm sure there'll be some pictures of me carrying my doctor hat. So, I'll let you know. So, let's see. Is there anything else about that? I think that's sort of the main thing. Again, people who've had my classes before know that sometimes you can get me off onto tangents, and I try and come back. So, yes, exactly. At least it's a good start, day one. Good. Today we'll see how far we get with this. The way I set up my lectures is usually, again, after the introductory section, is to have an outline of what I hope to get through, and then we'll see how far we get on that, particularly for the first couple of lectures, which are not individual virus based. Usually when we get to the individual virus or virus family basis, we'll get through pretty much all of that. And you'll see that that is set up pretty regularly. But today, much more of a general kind of approach. What are viruses? Turns out to be a much more difficult question than a lot of people thought. And literally in the last 5 to 10 years, it's become much less obvious what a virus is. Why study viruses? This is completely my reason to study viruses. It's not because they make us sick. It's because they're everywhere, and there's a ridiculous amount of diversity in them. And so we'll cover sort of that in a little bit more detail. History of virology. Actually, when people originally started studying viruses was because they were making people sick. A little bit about vaccines and pathogens and how these viruses were discovered in the first place. One of the problems with most viruses is they're just too darn small to be able to see. So virus detection and virus replication cycles turn out to be a very complicated and somewhat difficult way to look at things. So what is a virus, quote unquote? Well, again, it's a difficult question, so I'll obfuscate and move it to a little bit later on. Much easier to talk about is what's a virion. A virion is just the extracellular virus particle. So one of my definitions that I like to use is a bag of nucleic acid. So what is a virus? It's a bag of nucleic acid. It's a very specialized bag and very specialized nucleic acid. There are really pretty much two different kinds of virions. There's either the so-called enveloped virion or the naked virion. Enveloped is just because it's got a membrane around the outside, and naked has no membrane around the outside. And so clearly already a difference from cellular life forms is this extracellular, no membrane around the outside form here. There's also what's called the capsid. The capsid is just the proteins that make that bag. And so the capsid for a naked virus is just this proteins with the nucleic acid on the inside. Often people will talk about nucleocapsids. Almost all of these membrane-coated virions, the enveloped virions, have a separate protein which is either interacting directly or indirectly with the nucleic acid on the inside, and people usually call those nucleocapsids. And when we get to talking about particular things like HIV and the abbreviations, the horrible abbreviations we'll get to later on, anytime you see C, that's usually going to be capsid protein, or NC is going to be a nucleocapsid protein. But we'll get back to that again more and more later on. Most of these enveloped viruses, in fact, all the ones that I know of, all have proteins which are present in the envelope, and these would also be these are also known as the envelope or transmembrane proteins. Again, TM is a classic example of the name of a protein which you find in some of these virions. But why do I say this is not a virus, this virion? Because it's part of a life cycle that viruses go through. And one of my colleagues, Patrick Forterre, who's at the Institut Pasteur in Paris, came up with this, ceci n'est pas un virus. This is not a virus, it's just the virion. What's that reference to? Ceci n'est pas un pipe, René Magritte. So, virus definitions. Again, there are lots of them. The one that our textbook uses is that it's a very small, anytime Stedman puts quotes around things, air quotes or otherwise, means don't believe it. Infectious obligate intracellular parasite. Well, infectious and obligate intracellular I buy. Parasite, we can talk about some examples where I think of it as much more as a symbiosis rather than a parasitism. There are some nice examples of how viruses, in fact, work as mutualists, and in some cases even symbionts with their hosts. Why are they obligate intracellular? Because all viruses, quote unquote, need precursors, energy, translation, and membranes. Another definition is that viruses don't replicate through binary fissions. If you think about cells, you have one cell goes to 2 cells, goes to 4 cells, goes to 8 cells. For viruses, as we'll see, it'll be one virion goes to no virions, goes to many virions. And usually in a way which is a lot more than binary. 10, 100, 1,000 in some cases, you can get replication. Has a DNA or an RNA genome. There actually are a couple of examples where what's present, at least inside the virion, is both DNA and RNA. So HepDNA viruses that we'll get to right at the end. HIV actually encapsulates RNA, but 2 different kinds of RNA. And then, again, some of the work in our lab looks as if it may be, in some cases, a mixture of the 2 between them. And so these are sort of classic textbook definitions. My favorite definition is actually this one of Salvador Luria, and I'll let you all read it, but basically it's a very separate kind of definition which has to do with genomes being moved around. And nothing to do with, let's see, there's nothing to do with parasitism here. Using cellular machinery on the inside. It's a nucleic acid genome, and they can be moved around. So this is the one that I like better. I like the cartoon version. Here, this is what a virus is. The virus is not just that little green thing in A. It's A, B, C, and D. It's all this life cycle. So you have a virion with a genome in it. This virion associates with a host cell. That genome of the virus gets into the host cell. There's replication of this virus genome, sometimes a host genome, sometimes the host genome is completely gone. And then you make these extracellular particles, the virions. Those then get out and go through the whole process. And so one of the things that Patrick Forterre also came up with here is this idea of 2 different kinds of life. Of course, the virus life and the non-virus life. And how did he come up with the definitions here? He called the one kind of life, which is the cellular life, as the ribosome encoding organisms, or REOs. And then these guys, the leaders and rulers of this, the CEOs. Why CEOs? Because they're capsid encoding organisms. And that capsid is really unique to the virus. You don't have capsids. Cells don't have capsids. And so this is the real difference between the 2. So why do I study viruses? They're everywhere. We'll see some examples of that in about 2 or 3 minutes. Viruses infect everything. Plants, animals, fungi, archaeobacteria living in volcanic hot springs. Turns out that the exam we'll be going to in Finland has to do with some bacteria that infect hot springs, or live in hot springs, the viruses that infect those. Viruses are insanely diverse. That's not just because of their capsids, which are really diverse. Turns out the genomes are also incredibly diverse. And viruses are great tools for understanding the organisms they infect. And so again, a lot of what we know about molecular biology comes from studying viruses which are infecting these particular hosts. And yeah, sure, they'll make people sick. And these are my two favorite virus vectors here. This one's Helena, and that one's Sophia. This is a few years ago now. They're still getting bigger, but they're still doing a great job of moving viruses around. So yes, they infect my daughters. They infect me, but they basically seem to infect all of the organisms, cellular organisms that we found so far. For a while, people said, C. elegans isn't infected by viruses. Just a couple of years ago, they found some viruses that are infecting C. elegans. So pretty much any kind of organism that you can think of is infected. Unfortunately, at least for me and my funding, is that 99.9% of viruses that people study infect this group of organisms right here. And Homo represents the molecular diversity of all animals here. There are a few people who study viruses of plants. That's the other 0.1%. And the 0.001% are these viruses of archaea here. But every single one of these branches, which is from the small subunit RNA of the ribosome, all of these organisms have viruses that infect them. And if you look at just the organism itself, most of the times that organism's genome also includes viruses in it. And you can look at the human genome, depends on how you count, anywhere from 10 to 40% of the human genome is clearly viral. About what percentage of the human genome codes for proteins? About 1.5%. So we're basically more viral than we are human. That's kind of an interesting thing to think about. Some types of viruses are probably extremely old. This is, I think, an example of potentially one of these really, really old viruses, and we may get a chance to talk about this when we talk about virus structure a little bit later on in the course. I think that viruses were probably around at the origin of life and may have even predated that. I think the evidence for that is basically non-existent, but it's certainly there. Yeah?
I've heard you say that a few times, and what I always wonder is if they need a cell's machinery to do their thing, like, are they like reduced from, did they used to have the machinery? I mean, how do you think about that?
Right, so the question is here, just to record it, it's not as easy. How can I get away with saying they're obligate intracellular symbionts and being before cellular life? And so that's a great question. And one of the things that people argue about this, and again, I don't necessarily buy this, but people who make the arguments say that even if you had just an RNA replication, and just RNA replication by itself in the absence of cells, you see these parasitic replicators, which will take advantage of the replicating system which is already there, which may or may not be cellular. The other evidence for that is when you do simulations of what people think would have happened at the origin of life, you see these parasites that come up all the time. And so, again, what does it mean? If you were to go back into a pre-cellular world and look for viruses, then clearly the definitions here wouldn't work, as long as it's clearly an intracellular parasite. If it's just a bag of nucleic acid, okay, but you could also say that cells are bags of nucleic acid as well. So it's really a chicken and egg question. But I think we have evidence now that viruses were around at least when they were the last common ancestor of archaea, bacteria, and eukaryote. I think there's pretty good evidence for that, and that I definitely buy. Pre-cellular, yeah, we'll see. And clearly viruses are really important for evolution. One of the other quotes that I love to use is that we wouldn't have motherhood as we know it if we hadn't had a retroviral infection that changed mammalian genomes such that they could develop placenta. And I think there's really good data on that, and I'm happy to talk about that later. But the thing that really amazed me when I first heard about this is the absolute number of viruses, and really astronomical number of viruses which are out there. The first studies that were done on this were done by marine biologists who basically went out, took small samples of seawater, and then stained that seawater with dyes that bound to nucleic acid and fluoresced. And what they saw were a few, and actually very, very few, of these poor eukaryotes. There's actually one eukaryote right here. It's a diatom, which is bound to one of these nucleic acid binding stains. They saw a few more of these bacteria and archaea, which are these bigger dots in here, binding to the nucleic acid, but an absolutely ridiculous number of little spots in the background. And all of these are bags of nucleic acid, and probably almost all of them are virions. People argue about that as well. But this is, again, from an extremely small sample of seawater. It turns out you do the same thing for soil, you do the same thing for fresh water, you do the same thing for human blood, you do the same thing for pretty much any environment. You see absolutely ridiculous numbers here. So you do some back-of-the-envelope calculations, and you end up with 1 million to 10 million of these virions in ocean waters. And it's not just surface waters. It turns out that the viruses actually go down a long way as well. If you just look at these numbers here, 94% of the things that bind to nucleic acid binding stains here are those little dots, just by counting them. So there are more viruses in 1 cup of seawater than humans on Earth, and you multiply the number of milliliters of seawater on Earth versus the number that you see here. It's a absolutely ridiculous number. And this is a number that I don't think anybody can really comprehend unless you're an astrophysicist. Any astrophysicists in the room? Good. So none of us can comprehend it. If you would line up all of these virions from end to end, they'd be 100 million light years long, which again is a number that I can't deal with in any way. And what still blows me away is that this is in fact an example from my lab. And those people who work in my lab are not allowed to answer this question. How many viruses do you think are in this tube? And you can see my thumb up there at the top. Any guesses? 15 times 10 to the 7? A billion? Any other guesses? More than both of those? A bunch more than both of those? A whole bunch more than both of those? Yeah, this one's actually about 10 to the 11th virus particles in this tube. Which is, a billion is 10 to the 9. So that will be 100 billion in that one tube. A whole bunch more. And so this, again, to me, was sort of like, oh my God, this is, there are absolutely incredible numbers of viruses out there. Now this, admittedly, is a virus lysate from a cell, and a whole population of bacteria that were producing a huge amount of viruses. In these particular one, this FIX-174 virus is known to produce lots and lots of viruses. If you look at the human genome, a lot of you have seen this slide before. This gray segment here are the retroviral-like elements. These are the things that nobody would argue are viral derived. Again, it's about 8% of the genome. I would argue, and we can talk more about this later as well, that up to about 40% of the genome are viral or virally derived. These are the line elements, the sign elements, the retrotransposons that jump around in the genome, and of course, what makes us us is over here. Now, yeah, there are a bunch of RNA coding genes in there. Again, we can argue about that a little bit later. I mentioned before that the most diversity is present in viruses. Well, it's not just the diversity that you see in terms of their structures, which we'll take a look at in just a minute. It's the most genetic diversity. Few publications have come out in the last couple of years, although admittedly 2003, this one's getting a little old now. Number of people, particularly Roger Hendricks and Graham Hatfull and their group at University of Pittsburgh, but they now expanded. I don't know if anybody heard about C phages or looked at this. So it turns out if you go out and find a pretty common soil microbe, which is a mycobacterium related to Mycobacterium tuberculosis, but no, it doesn't give you TB. So these are mycobacteriophages. Go out and take any particular piece of dirt and look in there. You will find new viruses that nobody's ever seen before, which infect the mycobacteria. So a lot of people use this in freshman biology classes, some of the labs. We thought about doing it here as well. Just isolating those viruses, looking at them, and then sequencing them. They're up to about 4,000 different ones now. Some of them are called rosebush because they're found underneath the rosebush. Another one's called corn dog because it kind of looks like a corn dog under the electron microscope. If you look at the sequences of these viruses, and admittedly, thousands of these now have been found, 50% of those don't match any other sequence, the sequence of these genomes, anything else in the database, which is insane. But those are the really well-studied ones. If you look at marine viruses, like some of those little spots, and you isolate those particles, you sequence them, about 90% of those sequences don't match anything in the databases. And some of the viruses that we look at, 95+% of those genes match nothing in the databases. This is completely new sequence data. Where did it come from? How did it get there? Nobody knows. Really open and fascinating question. Again, you can do some back-of-the-envelope calculations. Billions of unique genes that are present in viruses. If you, for instance, however, look at a mammalian genome and try and see how many of those genes have been characterized, you will almost never find genes that haven't been found somewhere else. And if you do, they're viral genes. You can see really clearly that there's some viral containment, because again, this 10 to 40% of our human genome, a lot of the variability is actually in those genes, in those actual virus genes. And so people, again, back at the envelope calculations, very, very small numbers of what phage genomes have actually been characterized. We've even done some of this work as well. It's one of my students calls the dark matter of sequence space. If you just look at sequences that we found in an acidic hot lake in Lassen Volcanic National Park, 93% of the genes that we had in there matched absolutely nothing, and 7% matched virus genes, but most of those virus genes we had no idea what they were doing as well. And the reason this is called dark matter is because if you think about the universe, dark energy and dark matter end up being 96% of what out there, and actual visible matter is only about 4%. So again, astrophysics and virology, who'd have thunk it? But there's, I think, some really interesting parallels between what's known about these things and what's not known. Well, what do you see, however, if you now take those small dots, okay, fine, we sequence them, and it turns out DNA sequencing is actually really easy. Electron microscopy is a lot harder. But it turns out that, again, these are where you see the big differences at the structural level relative to, in fact, in parallel to some extent with what's going on at the sequence level. These things are completely different, absolutely and completely different than any cellular life which is out there. Many of them, as it turns out, have these structures which are icosahedral, and you will get really fed up with icosahedral virus structures, I will guarantee you, in the next couple of weeks. A few of them have these rod shapes, and it turns out a lot of these shapes have to do with just really basic, fundamental mathematical rules about how things fit together. The smallest surface area with the largest volume is what? It's a sphere. What do these guys look like? A lot like a sphere. And they're really easy to associate with usually relatively small numbers of proteins, which can sometimes be exactly the same ones in how they fit together. There's a lot more complicated virus particles, like this one here. This is a pox virus, some of the larger viruses which are out there. And this is an example of one of those corn dog-like viruses. See? This one right here. And this is just from finding those little spots that I showed you before, concentrating them, and then looking at them on the electron microscope. So these are weird, but these are way weirder. These are the archaeal viruses that we work with in my lab, and a few other labs in the world work with as well. These ones down here in the see your lower left-hand corner, the SSVs are kind of lemon-shaped viruses, and we've got now a higher resolution structure of these. This is one of my favorites, is the Acidianus bottle-shaped virus. It does kind of look like a champagne bottle with these little filaments at one end that almost look like candles on a birthday cake. This one here, the Acidianus filamentus virus, is twice as long as the poor cells that it infects and has these little nano-shaped claws at the end. And then I told you about this one this one here already. Why these guys are so different than each other, who knows? And that does bring me to a point, someone here was asking about whether that the finished defense is going to be filmed. That won't be, but I am giving a talk down in Corvallis Saturday at 1 o'clock. We're going to be talking about archaeal viruses, so I want a road trip down there. And that should be filmed. And I'm hopefully be able to take a look at that. So why study the viruses? Well, it's because they are really diverse. They're everywhere. Insanely interesting different structures, and a lot of the structures is really what me interested in them. But the sort of overall focus of the course, and again, getting back to Salvador Luria, is looking at unifying, simplifying, and overall patterns. And so that'll sort of be the focus of the course as we move along through the rest of it, looking for unifying, simplifying, and overall patterns. People will argue that my exams actually look for the isolated situations, but hopefully that's the main thing here. So a lot of open questions about viruses. Where do they come from in the first place? We already had that discussion already. How old are they? Why do viruses even continue to exist? If they're all parasitic and make us sick, you would think evolution would have gotten rid of them a long time ago. Because what's the selection for hanging on to something which is making you sick? And what that means is that probably the viruses that make you really sick are probably not very well evolutionarily adapted. So people call the virulent viruses, again, those that make their host really sick, are probably pretty new, evolutionarily speaking. Whereas the latent viruses, the ones that sort of hang out in the genomes, don't produce very much virus, don't make you very sick, may even in some cases make you well. It's the thing down here at the bottom. Are they good for you? They're probably a lot older, been around for a lot longer. Many viruses, and particularly a lot of those, again, the little dots that you see in the oceans, some of those actually carry photosynthesis genes which help their hosts grow. So the host actually grows better once it's infected by the virus than when it's not infected by the virus. And so that kind of brings up this, are viruses good for you? And this, Edge of Life, themovie.com, which is the link. It's also present on YouTube, and